All right, so we're going to be going into chapter 8, which talks about chromosome variation. Make sure that you go through chapter 8 in its entirety. I'm going to touch on important concepts throughout the chapter, but you need to go back and read it in its entirety. All right, we see chromosome variation in various organisms, and one in particular we're going to talk about is yeast. Yeast is it has a highly not yeast, I'm sorry, wheat. Wheat has a highly complex genome, and it is tied to evolutionary history, primarily because it is a hexaploid organism, meaning that it has 6 copies of genetic information, and these 6 copies of genetic information are derived from 3 different species. Scientists have done genomic sequencing that took about 13 years to complete, but it really gave rise to understanding hybrids, the development of hybrids, the evolutionary history and change and evolving of various organisms, but also the importance of understanding nondisjunction in accordance with mitosis and meiosis. So just looking at this example here, you can see that in the first generation, which we're going to denote as the P generation, okay, you have 2 different types of wheat that were crossed, okay. They produced this F1 generation, which was your first set of hybrids of wheat that was produced, okay. Went through a round of what we call mitotic non-disjunction, which we've covered, okay. And as a result of this mitotic non-disjunction, and just in case you don't remember about what non-disjunction is, it is the non-disjunction is the failure of one or more pairs of homologous chromosomes or sister chromatids to separate normally during nuclear division, and as a result, it results in abnormal distribution of the chromosomes, okay. So with that being said, after it goes through that F1 generation, goes through mitotic non-disjunction and it's bred, it produces more offspring, okay, that are then taken and are able to be crossed again. And this is new offspring producing another hybrid that now, as you see here, has 3 different sets of chromosomes, okay. And those 3 different sets produce what we call this hexaploid wheat. So it has a level of complexity to it that scientists have really used as a mechanism to further understand protein coding genes and how various repeats and modifying the genome can result in altered or have downstream effects. So let's just do a quick refresher. I talked about, when we talked about in the previous chapters, the chromosome variations, okay? In order for us to go into the mutations and understanding even non-disjunction a little bit further, we have to do a quick reminder on the chromosome morphology. Now, the foundation of the chromosome morphology is really based off of the position of the centromere on the chromosomes. That is the restricted area in the chromosome where, if you remember, we talked about the connector cores bind to, and it helps in the pulling apart of the chromatids or the sister chromatids during the process of mitosis and meiosis. Well, the chromosomes are classified, okay, based on 4 types, and the basis of them, their classification, is the presence of or the location of the centromere. So you have the metacentric chromosome, which is where the centromere is located in the middle of the chromosome, so you have 2 equal halves. Submetacentric, okay, is where you have the centromere that is displaced, or it's more on one end than the other, and so it creates a long and a short arm denoted as P and Q, okay, P being the short arm, Q being the long arm. And then you have acrocentric, and with acrocentric, the centromere is near one end more than the other, and so it produces a long arm and a knob or a satellite on the opposite end, excuse me. And then you have telecentric. Telecentric, remember, you can remember by the T, okay? It terminalizes, so the centromere is on the end, so there's not two equal halves or two distinguishable portions of the chromosome given the location of the centromere. Now, in order to look at or be able to identify the morphology, the size, the number of the chromosomes, you can develop a karyotype map, okay? Karyotyping is done when you want to identify a cell's chromosomes. You can actually take and harvest the cells from an organism. Usually you can draw blood, okay, and then you're going to take the cells that are harvested from that organism, and you're going to treat it with a substance that causes the cells to be frozen at metaphase or halted at metaphase. Usually one of the components is called colchicine, colchicine. Okay, colchicine. And what colchicine does is it causes the microtubules to be kind of frozen, and it halts them at the metaphase state. Well, what's interesting about the metaphasic state of a cell is that during mitosis, the cells are all distinguishably different because they are highly condensed, and they are lined up at the equatorial plane of the cell, at the middle of the cell, so you can distinguish one chromosome from another. And based off of that, you can then go in and begin to arrange the chromosomes based off of size. You can also stain the chromosomes, and that helps you to also identify one chromosome from another, and each chromosome will have a unique banding pattern based off of the stain that is used. For an example, you have the G bands, which the G stands for the GEMSA stain, okay? And this is a stain that allows one to distinguish the AT-rich region of the chromosome. The Q band uses a quinacrine mustard, okay? And that allows the chromosomes to be identified under an UV light, and you can see the varying brightness of the chromosomes based off of the presence of guanine and cytosine. Okay. You can also do C bands, which is centromeric heterochromatin, which is going to look more so or focus more on the centromeric region. And this is going to be also cytosine, guanine rich. And then you have R bands, okay, which is also cytosine and guanine rich. But ultimately, by utilizing the different varying stains and developing a karyotype, you can now see and distinguish one chromosome pair or homologous chromosome from another. Okay, so it's very, very helpful in identifying the varying chromosomes. This image right here is showing the chromosome, a karyotype of chromosomes within a cell that used GEMSA stain to see the G banding patterns. So when we talk about looking at the various chromosomes, it's important to be able to develop a karyotype because it allows you to see the state of the cell. Are the chromosomes that are present within the cell normal? Are there any abnormalities? And part of that helps you fully understand what's going on within the cell. Okay. Just like our DNA can be mutated by changing out bases, our chromosomes can be mutated, okay? Chromosome mutations can actually result in either varying, either the number of chromosomes or the structure can be varied. Either way, any of the type of mutation that is placed upon a chromosome can have a deleterious effect on the actual host cell. There are different types of mutations. There are three major types of chromosome mutations. You have your chromosome rearrangements, and this is where the structure of the chromosomes are altered. You also have aneuploidy. This is where the chromosome numbers are altered. Okay, so it doesn't have to mean in polyploidy where you have one or more sets of chromosomes that are added, so you have the addition of chromosomes. Aneuploidy can be where you have the removal or the deletion of chromosomes. So here you see chromosome rearrangement, and just to point out, this dot that you'll see is actually representative or indicative of the chromosome, the centromere. Okay, as you look here, you see that there are segments that are denoted by letters. Okay, and it helps you to understand, hopefully, the different types of mutations. So in chromosome rearrangement, you can have a form of like duplication. Okay, and we're going to go into this a little bit more. You also have aneuploidy, okay, or you can have polyploidy. In duplication or chromosome duplication, you have a mutation where a part of the chromosome has been duplicated, so it's been doubled. Okay, so you'll see here under the A section here, and this is about page 226 in your textbook, the chromosome segment has become duplicated. So as you'll see here, and you follow the arrow, you have this EF segment here, and it's been duplicated, and it's actually present twice on the chromosome. In a deletion, you have a portion of in the same EF segment that is no longer present in the chromosome, so it's been deleted out. In inversion, it's actually inverted. Okay, so it's turned into what we consider a 180 direction. So instead of it being EF, you have FE, so it's inverted. Okay, you see that here. And then translocation is where you have a segment of a chromosome that moves from one chromosome to a non-homologous chromosome. Okay, so it's moving, a portion of one chromosome is moving to a non-homologous chromosome, another portion. So here you see we have that EF segment. Okay, here's one chromosome, here's another. They're non-homologous, so they're not the same. And you have that portion of the EF that has now moved onto the another chromosome, and it's not homologous. They've almost switched. Okay, they've almost switched. Don't confuse translocation with crossing over, because they're not the same. Okay, it's not the same. So what are some effects of chromosome duplication? Okay, they primarily, you're going to have a major effect on the phenotype of that cell. Okay, so for an example here, when you look at this normal chromosome here, you have the normal chromosome, which goes from A to G. Okay, when you have chromosome duplication, you have that duplicated EF region that has been added. So now you're adding in multiple copies of, in this case, genes. Okay, so what can potentially happen is that duplicated region needs to loop out to allow the homologous sequences of the chromosomes to align, because if they're not looped out and removed, then you're going to have misalignment of the homologous chromosomes, which is going to be very important in meiosis when it's undergoing crossing over. We see this issue with effects of chromosome duplication in fruit flies, specifically the bar mutation. The bar mutation, it contributes to what you see, the shape of the eye. Okay, and so you see here in a normal situation, you have homozygous dominant, and it produces, you have 1, 2 copies of that bar region producing these normal shaped eyes. Okay, which tend to be oval. But in chromosome duplication, let's say if it's heterozygous, then you can have to where the shape of the eye has changed due to the reduced number of facets that contribute to the shape of the eye, and it makes the eye smaller and bar shaped instead of that normal oval shape that you see here. Okay, and when you have a homozygous bar female, which is what you see here, 2 Bs, okay, then what you have in this case here of this duplication, you get a further change in the shape of the eye, so less facets, and it's more bar shaped, and it becomes more exacerbated in a heterozygous double bar phenotype. And you can see that here. You often, just to point out, this is thought to be, or this is associated with X-linked traits, okay? And there are instances where the fly can carry, as you see here, multiple copies, like 3 copies of the bar duplication on the X chromosome, and then you have the impairment of the eye shape significantly reduced, and it also is thought to affect the other phenotypic outcomes as a result of that. Now, what is another downstream effect of chromosome duplications? Whether it's a duplication and or a deletion, they often arise from unequal crossing over. And as a result of that, the chromosomes misalign during crossing over, okay? You see this in color blindness, specifically with red-green color blindness in humans. The effect or the perception of color is affected by the red and green opsin genes, which you see here. The red opsin gene, the green opsin gene, okay? In this case here, the chromosomes don't align properly, and so you have unequal crossing over. The issue is, in a normal situation, excuse me, most people with normal color vision have one red opsin gene and one green opsin gene. Occasionally, you'll have 2 paired X chromosomes in a female that don't align properly in prophase 1, and it can lead to unequal crossing over. Okay, when you have the unequal crossing over that occurs, it will produce 1 chromosome with an extra opsin gene and 1 chromosome that is missing the opsin gene, and that's what you're seeing here. Okay, so 2 here, and then 1. And then there's 1 on this opposite chromosome, but the other is missing, okay? So unbalanced gene doses. So what happens when you have what we consider to be an unbalanced gene dosage? The amount of, and this deals with the amount of protein or product that is produced from the genome, okay? We see this primarily during development. And the issue that arises from this is that the amount of protein that is synthesized by a cell is often directly related to the number of copies within a cell. However, if you have more copies than in a cell than you should, it will then produce more protein. So the downstream effect is more genes, more proteins, okay? The issue with producing more protein is that it can cause have downstream effects in the cell itself. Remember, the cell is always trying to stay at a state of homeostasis. So there is an instance where if you have too much protein or not enough, it can cause abnormalities. And this is what can happen when you have unbalanced gene dosage, which you see here. Okay, so as you see here in this instance with the transitioning from the embryo to the fish, development process often require the interaction of various genes. In this case, the examples is A, B, and C genes. Okay, and the interaction of these gene products have a downstream effect in the process of development and producing what we would consider a normal fish derived from an embryo. Okay, however, when you have duplication and other types of chromosome mutations, you can either produce multiple copies or extra copies of some of the genes. And as a result, when you have extra copies of the genes, in this case here is an example of the B gene, then it's going to alter the amount of product that is produced. And as a result, the more product can result in abnormal development. So you see here, there's one stripe or pattern on the fish But when you have extra product produced due to gene duplication, then you can have an abnormal phenotype or morphology. So how do you get abnormal gene duplication or abnormal chromosome numbers? It is derived from various reasons. You can have chromosome deletions, okay? And this is another type of arrangement. This is where you have a loss of a chromosome segment. A large deletion is usually more easily detected than a smaller deletion. But either way, you can have any type of deletion or loss of chromosomes can have a deleterious effect. Some of the effects of these deletions is imbalance in gene product. When you have an imbalance in gene product, that means you can have not enough protein to carry out a certain function. And not having enough protein, which is the product of the gene, can result in the inactivity of, let's say, enzymes or proteins that are necessary. You can also have the expression of a normally recessive gene, which we call pseudodominance. And this derives from having a gene that would normally be recessive that can be deleted and have a downstream effect of lack of product produced. And then haploinsufficiency. Haploinsufficiency is when you have an instance where two forms of the gene are required to produce sufficient product to carry out a certain task. But in haploinsufficiency, just producing one, for an example, let's say one form of it is deleted because of a mutation, then the half or whatever is present is not enough. The product that's produced from that gene is not enough to produce the wild type phenotype. It is considered haploinsufficient. We see this with in fruit flies with the notch mutations. So in this instance here, we see an inversion, okay, due to an issue or abnormality in meiosis. And when you have, in this case here, you have an individual that is heterozygous for a deletion, and the normal chromosome has to be looped out, as I showed you guys earlier, so that you can have proper alignment of the chromosomes. Because if you don't have proper alignment of the chromosomes, then you're going to have misalignment, and it'll impact normal crossing over. So this has to do with, or leads us to, chromosome rearrangements, because there are instances in the cell where you have chromosome abnormalities due to deletions or duplications. And so if there is something that causes a genetic abnormality in the arrangement or the localization of these various genes, it has to be corrected. In the last instance we saw, there was a looping out that had to occur to allow for proper alignment. Okay. There are also instances where you have what we consider to be inversions. This is the third type of chromosome rearrangements. And this is where a segment of the chromosome is inverted in that 180 degrees that I showed you guys earlier. Okay. This is also impacted by the presence of the centromere. If the inversion includes the centromere, it is a paracentric inversion. If it does not include the centromere, it is a paracentric. Para is P-A-R-A. Okay, paracentric is when it does not include the inversion, the centromere. And the inversion that includes it is P-E-R-I, centric. And you can see that here. This is how it would normally look in the chromosomes A through F, with the centromere being in between C and D segments. But in pericentric, P-E, okay, the inversion occurs within or includes the centromere. Okay, there's your centromere. And paracentric, you have the inversion that occurs outside of the centromere, so it does not include the centromere. Okay, we see this, and this was an example of this is with in chimpanzees on chromosome between the human chromosome 4 and the chimpanzee chromosome 4. So if the inversion occurs in inversions and meiosis, it's between individual homozygous, then no problems arise during meiosis. Okay, so no problems arise, and the two homologous chromosomes can pair and separate normally as you would expect to see in meiosis. However, when an individual is heterozygous for an inversion, the gene order of the two homologs differ. And as a result, the homologous sequences can ally and pair only if the two chromosomes form an inversion loop. And that's what we saw, what we can see right here. Okay, the inversion loop. And this is during prophase 1 of meiosis. A visual example of what I was just talking about in paracentric versus pericentric, this would be the example. Okay, so in A, you have what we consider to be normal or wild type. Okay, and you can see the inversion here. Okay, so in prophase 1, the inversion loop forms and a single cross within the inverted region. So you have crossing over within the inversion. And it results in this abnormal or unusual structure that you see here. In anaphase, the centromeres are going to separate, stretch the, what we call the dicentric chromatid. Now, a dicentric chromatid is where you have one of the four chromatids, that have the centromere. Okay, so when you have one of the four chromatids that have two of the centromeres, then you call it a dicentric. So it's one of the four chromatids that have two of the centromeres. That would be a dicentric chromosome. Okay, and that also leads to the dicentric bridge that you see right here. Hopefully it's showing up in the PowerPoint. Okay. The two sister chromatids in anaphase 2 separate and form the four gametes, which you see. Okay. Two of those gametes contain the non-recombinant chromosomes. One is going to be wild type. One is going to be the one with the inversion that we just saw up here. And then the other two recombinant chromosomes are going to be missing some of the genes, and the gametes probably will not produce viable offspring. And that's really going to be determined by if that region has lethal genes or not. Okay, but two of them may not produce viable offspring. This is pericentric, the same example, but with pericentric inversion. All right, so again, I want to reinforce the concept of not confusing translocation with crossing over. Translocation includes the movement of genetic material between non-homologous chromosomes. Okay, when we're talking about crossing over. Crossing over should be occurring between homologous chromosomes. And remember, in the instance where it's not homologous between like the sex chromosomes, it still has homologous regions, the regions that allow for that necessary crossing over. Okay. But with translocation, you can also have it occur, that movement of genetic material in homologous or same chromosomes. So it's not just specific to non-homologous chromosomes. It also can be with non-homologous chromosomes, but it should not be confused with crossing over. So here there are three types that we're going to cover quickly, and that is non-reciprocal translocation, reciprocal translocation, and Robertsonian translocation. So you can see here in reciprocal translocation, there is a two-way exchange, okay? 1, 2. There's a two-way exchange of segments between the chromosomes. In non-reciprocal translocation, okay, you'll see here that the genetic material moves from one chromosome to another without any reciprocal change. So there was no reciprocal change between the two. And the third type is Robertsonian translocation, okay? And this is where the long arms of the two acrocentric chromosomes become joined to a common centromere through translocation. And it generates a metacentric chromosome with two long arms and another chromosome with two very small arms. Now, oftentimes with Robertsonian translocation, that smaller chromosome is usually lost. And the reason why it's lost is because it doesn't have enough mass to segregate properly during mitosis and meiosis. And so ultimately, as a result of Robertsonian translocation, you have the loss or reduction of chromosome number. Okay. In this case here, where you have reciprocal translocation and meiosis, you'll see where you have an individual. We'll start from the top, from A. The individual heterozygous for translocation is going to possess one normal copy of the chromosomes, denoted by N1 and N2. Okay, and then it'll also have a translocated copy, T1 and T2. Because the sets of chromosomes that are homologous, okay, they form together during this translocation process in what we call a cross-like configuration. And this is in prophase 1 of meiosis. So it forms this cross-like structure here. As a result, in anaphase 1, the chromosomes are going to separate in 3 different ways. You can have what we call alternate segregation, okay, where you have N's together, T's together. You can have adjacent, okay, where they are adjacent to each other. Or you can have adjacent to, which is a more rare form of translocation. But in this case here, it actually is the N1 and N2 move toward one pole, and the T1 and N2 move towards the opposite pole. Either way, in both of these 2 examples right here, the adjacent 1 and adjacent 2, the centromeres segregate to opposite poles. So you can see that the centromeres segregate to opposite poles. So what this shows is that gametes resulting from the adjacent 1 and the adjacent 2 segregations are non-viable, because some of the genes are present in 2 copies, where in the other copies some of the gene products are missing. And this is more prominent with lethality. Okay, we talked about lethality already. There are some chromosomes that contain what we consider fragile sites. This is where you have sites that develop constrictions or gaps when the cells are grown in culture, and they're prone to breakage under certain conditions. There are more than 100 fragile sites on the human chromosome, okay? And those regions are highly susceptible, or more susceptible, to breakage. One of the examples is the Fragile X syndrome. And this is a disorder that includes intellectual disability. And the individual with Fragile X syndrome exhibits X-linked inheritance, and it rises with a frequency of about 1 in 5,000 male births. And it's shown to be associated with the increase in the number of the CGGG repeats. So let's briefly go into aneuploidy. We kind of already started to cover aneuploidy, but I want to go over it a little bit more. And aneuploidy is referring to the abnormal number, or the variations in the number of chromosomes, okay? It can be an increase or a decrease in the number of individual chromosomes, okay? Anytime you have a variation in the copy number within a cell, it's referred to as aneuploidy or polyploidy. So what is the cause of aneuploidy? At least three potential causes. It can be from the loss in the course of mitosis. It can be lost, or the chromosome content can be lost during mitosis or meiosis. For example, the centromere becoming deleted. Another example is the small chromosome that was generated in the Robertsonian translocation can be lost. We talked about that. In Robertsonian translocation, because you have an imbalance in the or unequal distribution or transfer, what happens is a small fragment or chromosome is produced, and the mass of it is not strong enough to withstand the process of division, and so it becomes lost. And the third example is can arise through nondisjunction. Remember, we talked about nondisjunction, and it can occur during nondisjunction and meiosis and mitosis. So you see here, it's showing you how these aneuploids can be produced through nondisjunction in meiosis 1, meiosis 2, or mitosis. So you can see here, they show you the nondisjunction in meiosis 1, where you have an abnormal number of chromosomes due to the unequal separation. You see that here. Okay. And as a result, it produces, in this case, the zygote is trisomic. Okay. Or you can have it in meiosis 2. So normal meiosis 1 is normal. Meiosis 2, you have this nondisjunction here. So one cell does not have the proper chromosome number. And as a result, you have trisomic here and monosomic here. So an extra chromosome here, missing a chromosome here. And then in this case, in nondisjunction and mitosis, the nondisjunction can result in the somatic cells where there are some that are monosomic, or they can be trisomic, which they have that extra chromosome. Okay. So what are the types of aneuploidy? So you have nullisomy, and this is the loss of both homologous pairs of chromosomes. And it's represented, as you see here, by 2n-2. You also have monosomy, okay, or monosonic. And this is the loss of a single chromosome. So mono being 1, okay. Trisomy, tris 3, so you have the gain of a single chromosome. And then tetrasomy is where you have the gain of 2 homologous chromosomes. So what are some of the effects of aneuploidy? Examples, one in plants, okay? In plants, you see this in the seed cases in the Jimson weed. And as a result, it produces different phenotypes because of the varying number of the chromosomes, which you see here. Okay, so this is what it should normally look like. Okay, but these are the varying types based off of the aneuploidy. Okay, so that's one example. But then we also have aneuploidy in humans, and we talked about both of these syndromes already. Okay, Turner syndrome, where you have the absence of one, and then Klinefelter syndrome is where you have more than one X chromosome. Okay, so these two examples are where you have aneuploids in humans. Okay, and we also see that this can be present in what we call autosomal aneuploids, and this is something that many people are familiar with, and that's trisomy 21, which is Down syndrome. Okay, you have primary Down syndrome, which 75% random non-disjunctions in egg formation, and then you also have familial Down syndrome, and this ties back to the Robertsonian translocation between chromosomes 14 and 21. Okay, 14 and 21. Most children with Down syndrome are born to unaffected parents, and the failure of the chromosomes to divide has little heredity tendency. So if you have a couple who has conceived one child with primary Down syndrome, they have only a slightly higher risk of conceiving a second child with Down syndrome. About 4% of Down syndrome, people with Down syndrome, are not trisomic for complete chromosome 21, but rather have 46 chromosomes, but one extra copy or part of chromosome 21 that is usually attached to another chromosome through translocations. In familial Down syndrome, this tends to be inherited because it runs in the family, and the phenotype or the symptoms are similar to that of primary Down syndrome. Okay, so this would be the example of Down syndrome here. So in this case here, you can see the aneuploidy of chromosome 21, which is here. Instead of having 2, there are 3. And usually the phenotype of an individual with Down syndrome, they typically have very similar features, and it's usually also associated with intellectual disabilities and abnormal facial features, like eyes, flat face, small nose, oval shaped eyes. Okay. It is believed that you have the translocation of chromosome 21 onto another chromosome, and it results in familial Down syndrome. So between these two here. Okay. So this example here is showing you the aneuploidy carriers of translocation for chromosome 21 and actually how this can occur. Okay. This is very helpful. I won't say critical, but very helpful in understanding trisomy 21. Okay. So here you have the P generation, normal parents undergoing gametogenesis, and then you have the F1 generation. Okay. Now, a parent who is a carrier for 1421 translocation has a normal phenotype. Okay. But through gametogenesis, they produce gametes in these possible combinations that are denoted here. Okay. And as a result, they have about 1/3 of the children, the possible gametes, that can produce Down syndrome or have Down syndrome. The others that do not become full zygotes will actually be aborted in embryo. So outside of having trisomy 21, there are other autosomal aneuploids that are a result of aneuploidy and translocation in humans. You have trisomy 18, which is called Edwards syndrome, and it affects about 1 in 8,000 live births. You have trisomy 13, which is Patal syndrome, and that's about 1 in 15,000 live births. You have trisomy 8, which is a little bit more rare than the others, and this is 1 in 25,000 to 1 in 50,000 live births. Okay, so ask yourself the question, and I want you guys to look this question up. It's in the textbook, about page 245, 244. But why is there a drastic decrease in the frequency of this trisomic syndrome from chromosome 18 to 8? I want you guys to look it up. You may see this question again. Okay. Why is there a drastic decrease from 18 to 8? Okay. All right, we're going to stop here. And